receptors or stretch receptors. So as air moves into your lungs and the visceral pleura is stretched and the muscles are stretched, then sensory input coming in from the mechanoreceptors will cause the expiratory neurons to fire, okay, if you inhale. So you normally aren't using the ventral respiratory group in just quiet breathing. So when you were just trying to set your tidal volume on the biohack last week, and you were just doing the 5 normal breaths, you were just involving the dorsal respiratory group. All right? Then we are doing increasing your inspiration and forced exhalation. So we went to do our forced expiratory volume, the FEV1. And just breathing out as hard and as fast as possible, you are involving the ventral respiratory group for that activity. And the dorsal respiratory group for just the normal tidal volume, but forced changes to that, you're going to involve the ventral respiratory group. So expiratory neurons here are doing our active expiratory muscles. So the internal intercostals and the anterior abdominal muscles. So forced inhalation and expiration. And these are going to go to internal intercostals and abdominal muscles. So here's a diagram of the ventral respiratory cycling, okay? So if you do active expiration, then you involve the expiratory center of the ventral respiratory group. That's the more detailed portion of respiration. When we look at the pontine respiratory group, now we're looking at fine-tuning. Okay, both the pneumotaxic and the apneustic fine-tune the respiratory rate. So here's the key thing on this slide as far as the pneumotaxic group does. Okay, it increases the respiratory rate by itself, usually involving changes from the higher cerebral center. So the chemoreceptors are going to go directly to the dorsal respiratory group, but involving emotions is going to affect the pneumotaxic group, okay, as far as changing your respiratory rate. So breathing with the pneumotaxic group is more of a panting type of effect. If that is cut off, so it's not available, so if we do a transection across the pons right here, okay, you're still going to breathe, obviously, because we still have the medulla, but you start breathing. So it prolongs the inspiration and you have short expiration. With this being active, it's the inspiration is shorter, all right? So with the pneumotaxic group, you have a short inspiration. Without it, you have prolonged. Normal is 2 seconds in, 3 seconds out. So if it's not active at all, you go. And if it's turned on all the time, if it's more active, then it's. I have to stop doing that or I'll pass out. Okay. Okay, apneustic kind of does the opposite of the pontine. It prolongs the deep inhalations. Remember, if we didn't have the pontine, we go. So that's what the apneutic does, and it prevents the cessation of breathing. Okay, so it may be a ragged breathing, but it prevents the cessation, but still a fine tuning. So often the pontine centers will combine both of these together. This looks really complicated, so let's go through it slowly because it's more of an interesting thing than a complicated thing. This is what it's meant to be. All right. So we're looking at the medulla. This would be the respiratory center in the medulla. This is the pons, and up here is the midbrain. Everybody with me on that? All right. So if we cut below the medulla, so if we cut in this area here, then there is, let's see if I can do this on here. I'm going to erase my wrap effect. So if we cut below the medulla, no breathing. Because we need the respiratory center to breathe, okay? So that's the respiratory arrest here. If we don't have, remember what the breathing centers and the pons are for? Fine tuning, okay? So if we cut below those centers and we have just the medullary center, and we cut here, then we have this, you're breathing, but it's irregular. We don't have the fine tuning of the pons, all right? So if we cut here, then we have gasping. So breathing is occurring, but it's irregular. If we have medulla, but not pons. And if we don't have the pneumotaxic center, right, we still have the apneutic that's going to keep us breathing, but if we don't have the pneumotaxic center, recall this previous slide, we would have the prolonged inspiration with short gasping. So we'd still be gasping. Would it be like this? Okay, so long inhalation, brief expiration, or exhalation. And then if we cut up here, normal breathing. You don't have to worry about with the vagus or without the vagus. That's affecting the speech. So I've kind of written it back out here on this screen. So that's what we've covered so far. Let's just recap a bit here. What we've covered so far now is what the breathing looks like, not what's driving the breathing other than our pattern generator. So after I recap this, then we're going to look at these chemoreceptors. Okay? So, let me recap this before I change the board. We have two areas of nuclei in the brainstem that affect how deep we breathe and the pattern of breathing. We have the respiratory center in the medulla, and the 2 areas in the pons. These are required for breathing. The medulla, again, is required for breathing. If we don't have that at all, we don't breathe. So there's nothing in the spinal cord, even though that's where our lower motor neurons are, there's nothing in the spinal cord to trigger that. Okay? We can't even get voluntary control down to the spinal cord if we have a transection. So you can't mentally say, I'm going to take a breath. I'm going to stop that, because any upper motor neurons coming down to the diaphragm would be transectioned as well. Does that make sense? So both the voluntary and the reflex control are abolished if we cut below the medulla. All right, the dorsal respiratory group is our inspiration neurons for normal tidal volume breathing. The ventral respiratory group is what we bring into control when we're doing forced inspiration and forced exhalation. All right. So again, back to our FEV1, FEVC, when we were measuring that, that's going to involve the ventral respiratory group. These are activating skeletal muscle. So breathing is a voluntary muscle, skeletal muscle type reflex. The patterns of inspiration and expiration are modified by the pneumotaxic and apneustic centers. Okay. So primarily, inspiration is shortened if we use the pneumotaxic. Without the pneumotaxic, inspiration is very prolonged. Okay? So again, the primary slides that I utilized for those lecture notes involve.
I'm just confused really quick.
Yeah.
If you cut up there, wouldn't you not have any of that?
If you cut up where? The top?
The top.
You wouldn't have voluntary. So in other words, in Bio 430 we talked about marionecephallic babies that were born without a cerebral cortex, basically enlarged ventricles with very little gray matter on the cerebral cortex. So, but their brainstem is intact. So you still have, if we cut above the medulla, you would still have the pre-Botzinger complex, and you would still have the dorsal respiratory group. So you would have that irregular, you would still have the gasping. You include the pons, which most marionecephallic babies do, you would have that breathing shown up there with the long inhalation components.
So the bottom part will work if the top part is not connected?
Yeah. So you wouldn't have you wouldn't be able to take a deep breath on purpose, but you would still have the breathing occurring, because that's a reflex, which we can override. Just like I have a reflex for my knee jerk, if I have a transection of my spinal cord, that knee jerk reflex will still work. Okay, I won't have the voluntary extension of my knee, but the knee jerk reflex in response to tapping of the patellar tendon would still work. All right, so the generation of our rhythm, what the dorsal respiratory groups and the ventral respiratory groups do. This is just more detailed information on those groups. And then this diagram here that I just left on the board about what, reinforcing what they do by the changes that occur with the cutting. So now let's look at the chemoreceptors, all right? How do we then modify this basic pattern? We have chemoreceptors present in the central nervous system, and we have chemoreceptors present in the peripheral nervous system. Oh, I have it here on our left. One second. 78. May I raise the board? You guys, just give me some board. So redraw the pons and medulla over here. And hopefully you kind of recognize these vessels. So there are chemoreceptors present in the medulla, known as CNS. Chemoreceptors that are associated with our ventral dorsal respiratory groups. There are chemoreceptors located here known as the carotid bodies and aortic bodies. So notice this is arterial blood that we're looking at here. The neuron, which is sensory to the carotid bodies, is the glossopharyngeal nerve. And the aortic body is vagus. So we're looking at the amount of carbon dioxide, the level of hydrogen ions, and the amount of oxygen as a gas dissolved in the plasma. So we're looking at the central nervous system and peripheral nervous system components. So central nervous system is going to be affected by arterial PCO2 and arterial hydrogen ions. Oxygen is not detected or affecting our central nervous system chemoreceptors. But that is going to be a factor of our peripheral nervous system chemoreceptors. So we have arterial hydrogen ions, arterial PO2, and arterial PCO2. So as we look at this diagram up on the board, all right, this is cerebral spinal fluid. So where would we have cerebral spinal fluid?
In the cerebral
We're
spine.
going to have it in the fourth ventricle.
On there,
And we're also going to have it in the subarachnoid space. Remember? So if we have pia mater covering the surface of the brain, and then have arachnoid matter. This would be our subarachnoid space. And it's also going to have CSF in it. This is our fourth ventricle. So our cerebral arteries, in the medulla we're talking about branches of the vertebral arteries. Remember the two vertebral arteries travel along the medulla to join on the basal pons to form the basilar artery. So we have our vertebral arteries on the medulla sending out branches. Our posterior pica, we talked about last semester, if you remember that. And those are going to form arterioles and small capillaries and penetrate the medulla to provide the neurons with oxygen and nutrients. And also, fluid is going to get into the, not blood fluid, it's going to go by the CSF. So in our blood vessel here, which has obtained its blood from our cerebral arteries, it has both carbon dioxide and hydrogen ions. The two things that I said here that are sensitive in the CNS chemoreceptors. Well, I'm going to put parentheses around our arterial hydrogen ions because as you can see here, hydrogen ions cannot exit the capillaries due to the blood-brain barrier. However, carbon dioxide can because it is a gas. So carbon dioxide diffuses into the CSF. And present in the CSF, we have a familiar enzyme, carbonic anhydrase. Remember that speeds up the rate of the reaction. It occurs slowly anyway in the plasma, but this speeds up the rate of the reaction and forms carbonic acid, which then can just dissociate to form hydrogen ions and bicarbonate ions. So now we have chemoreceptors for hydrogen ions along the fourth ventricle. And as those bind to these hydrogen ions, they send signals to our dorsal respiratory group. So these are receptors, so they're going to have graded potentials. And more of those graded potentials are going to form as hydrogen ion concentrations increase. So indirectly, the arterial PCO2 is going to increase hydrogen ions and end up activating the dorsal respiratory group so that it has a more frequent pattern of the ramp. So you're going to have a shorter 3-second expiration and a more frequent, probably still close to 2 seconds inspiration. And also this can become deeper, not just more frequent tidal ventilation, but more deeper ones as well. So that's the primary role of the central chemoreceptors. So when you're rebreathing the paper bag, your own air from the paper bag or the plastic bag, you're breathing back into it, you're breathing higher and higher levels of CO2. Remember, normally we have about 4% CO2, a CO2 of ranging between 40 and 45, depending on whether we're exhaling or inhaling. And now as that builds up, what's going to happen to the CO2 in your blood as it goes by through the pulmonary capillaries? It's at 45 millimeters per mercury, PCO2 in your plasma. The alveoli get to 45, and then to 46, and then to 48. So the CO2 that's coming into the lungs from your venous blood is not going to be able to diffuse up a gradient, and in fact, it's going to stay in the plasma, and as the CO2 in your lungs rises, it's actually going to start adding to that. So now as the blood leaves the lungs and returns to the heart in the pulmonary veins, and then is pumped out of the left ventricle, it's going to end up with a higher level of PCO2 instead of a normal. Right? When that blood then gets to the brain, higher levels of CO2 are going to form more carbonic acid, which forms more hydrogen ions, which triggers greater numbers of action potentials, greater potentials rather, to the dorsal respiratory group, and it fires more often. And it may even involve the ventral respiratory group if we breathe more deeply. Okay, opposite. F. You're going to hyperventilate. Hyperventilating is not just breathing faster, but breathing more deeply. So you're blowing off CO2. Do that for 3 minutes. I'm going to get a little dizzy. Okay? So that when you do this in lab, please be sitting down. And if you start to feel dizzy, stop doing it and put your head down between your knees. Okay? You should have a partner working with you. So I don't want you standing up doing jumping jacks while you're doing the hyperventilation. So what are we doing now to levels of CO2? Maybe now the blood coming into the pulmonary capillaries, instead of being 45, is 40 or 38. And so then we have less CO2 as it goes back to the brain. So what we're signaling our dorsal respiratory group is, normally let's say it fires, with a set level of CO2, and now we have less, it's going to fire less often. So you take fewer breaths. You may not even take a breath at all if the CO2 levels are low, until they build back up in your blood, because you're not breathing. It's going to build back up in your blood, and then finally it's going to trigger a respiration again. Okay? So my dad pulled a trick on this with this when he was in college. One of his friends was trying to advertise a ROTC type medical club, and got my dad to play in with him. So during chapel, college hour, he said, I want you to hyperventilate and pass out. Well, my dad had just gotten through playing soccer and gotten a pretty bad hit, and he was sitting next to his friend who had given him the hit. And so during chapel, we started hyperventilating and passed out and slid right off the chair. And the poor friend was scared to death that it was a side effect of the hit he had gotten. And the guy advertising the club just wanted to come over and do some pretend CPR and kind of thing and get some interest in his club. But he scared my dad's friend quite badly. So this is the main effect that we're working with in lab today when we do the hyperventilating or rebreathing. Okay? Now, let's switch over to peripheral nervous system. So here we're looking at pH, PO2 changes, and PCO2. You notice the order that I put them in over there under P and S? So the primary one is blood pH, hydrogen ions in arterial blood. Next order of importance is oxygen, PO2, but typically not unless it drops below a PO2 of 60. Normally our arterial PO2 is around 104. So this doesn't really become a factor in most individuals unless it drops below 60. And then of less sensitivity is PCO2. So we're talking about the aortic bodies and the carotid bodies, all right, are most sensitive to arterial hydrogen ions. In contrast, remember, blood-brain barrier keeps the hydrogen ions in the arterial blood from exiting the vessels. Here we have direct sensors for those in the blood vessels themselves. All right, so here's an image of the carotid body, called the carotid glomus. It's a really cool, every time the teacher says that, students cringe, right? So here's how the glomus body works, and this is the last content of note that I want you to know, okay, is this slide. So this is a glomus cell, this whole thing right here, and under, Jeff was visiting with me in my office yesterday. We don't really have oxygen sensors, receptors for oxygen, I said, well, there's changes in iron ion content, and then we have these